should we decide whether some being is alive or dead? How does one actually define life? There have been and are really two quite different approaches to that question, which I'll call the metabolic and the genetic. The metabolic approach is to say that a living organism has some kind of fixed form and structure. It may change gradually, but it has a form. But all its components, all the atoms and molecules that compose it, are coming in from the outside, are being changed, and are leaving. So it's a constant structure with a continuous flux of material through it, requiring energy for its maintenance. It's a kind of thing, I nearly said the kind of nonsense, that biochemists are liable to say. It has, if you think about it, to be nonsense. If it were true, that flame would be alive. The flame has a clear structure, with a blue center and a mauve surround. Yet every atom is only part of it for a fraction of a second, I guess. You may say, well, it isn't alive because if you turn the gas off, it would stop. But let's face it, I could say you're not alive because if I deprived you of food and oxygen, you would go out. The fact that it's being fed by gas proves nothing. It has another property of life, if you think about it. It reproduces. That Bunsen burner was lit by a match, by another fire. We could, though I promise I won't, use it to set light to something else on the bench here. I mean, in other words, fire reproduces. It has all the metabolic properties of life, and yet, clearly, in some sense, it's not alive. What is it that it is missing? The other view, which I would call the geneticist view, is that flame was not alive because it lacks heredity. It lacks the property that like begets like. And that, in fact, the essential properties of living things are that they have three properties. Multiplication, which the fire has, that 1 will give rise to 2, 2 will give rise to 3. It has variation. Well, flames have variation. I mean, they can be a house on fire or a Bunsen burner, which have very different forms. But it must also have heredity, and that is what the flower does not have. And I've described heredity in this slide. There must be multiplication. An A must divide and give rise to 2 offspring. A B must divide and give rise to 2 offspring. But when multiplication takes place, A's must give rise to A's, and B's must give rise to B's. Just occasionally, perhaps, a mistake happens, and an A gives rise not to an A, as it, so to speak, ought, but gives rise to a C. But if it does, then the C will, as we say, breed true, will give rise to C's. That is the property of heredity, and it's the property that the flame lacks. The nature of that flame does not depend upon whether it was lit by a match or by a cigarette lighter or by another Bunsen burner. Its parent doesn't affect its nature. Its nature is entirely determined by the immediate environment and not by its history. Why do I hark on like this about heredity? Why does it matter? It matters essentially for the following reason, that if you have entities which have these three properties, multiplication, variation, and heredity, then quite necessarily you will have evolution by natural selection, and you get everything else in a sense for free. I mean, what we really want to understand are not the molecules I'll be talking about later on, but we want to understand primroses and elephants and people and fruit bats and things. We want to understand really complicated objects. But in a sense, that's not a difficulty. Once we've got natural selection, the rest follows, and you can't have natural selection without heredity. So that's why this talk is really, although I've called it the origin of life, is actually going to be about the origin of heredity, because that's what the hard thing to explain is. The question is where to start. And I want to start by the notion of crystallization, though I don't think crystallization is heredity for reasons I'll explain in a moment. What we have here is a solution, a supersaturated solution, which has no crystalline structure. It's just a pure liquid. But when Bryson adds a crystal to it, as a sort of seed. With any luck, it will crystallize, and indeed it is. It's gradually, the crystal is growing, new crystals are appearing, and give it another few seconds, and that solution will have crystallized. In that sense, it's rather like a fire, that when you give it, you can light it in a sense, but this time you light it with a crystal, more crystals will grow, and when it's crystallized, we could then use one of the crystals from there to seed another test tube of supersaturated solution. That would crystallize, and so on. But as yet, there's no heredity. Now, how does crystallization work? It will become obvious to you as this talk goes on that I am not a chemist. I do not understand chemistry. Somehow or other they failed to teach me any chemistry when I was a boy, and I've never quite caught up on it. But nevertheless, the principle is like this. I hope. You have in the solution some objects, which I've called monomers, some, molecules, and they have bumps and holes on them, holes like that and bumps like that. And the rules that I've made up for this particular crystal is that two of these monomers will not stick together unless at least two of the little pegs of one of them can stick into at least two holes. If it can, then it'll stay together, but one bond, as we call it, isn't enough. And if you look at them, there's just no way two of them can join together with that property. In order to get the thing to crystallize, you have to put in a seed like this, which you've put together in some way, or which maybe happened by the really rather rare chance event that sometimes happens in the world. You get a seed. Once we've got that, there's a slot there that one of these can fit into, forming something. Here he is fitted in. Once he's fitted in, there's a slot here that that one can fit into, and then this one, and then that one, and the thing will go on growing. And if we added another rule that when it got to a certain size, it's likely to break in half, then we have got reproduction. We've got an object which replicates. But it isn't actually heredity, because there's only one kind of object you can have. There's no variation. And you can't have heredity without variation. If there's only one kind of thing, you can't say that like begets like in any meaningful sense. But it turns out that you really can make a very simple modification to that kind of scheme, and you've got heredity. And there it is. I've now got two kinds of units, which are coloured differently, called A and B. They happen to be mirror images of one another, but that's not logically necessary. And they, too, will not crystallize on their own. To get them to crystallize and form a polymer, you have to put in a seed. But now there are two kinds of seed you can make, both containing two B's and an A in this case, but fitted together in different ways. If we seed the solution with one of those, it'll build a polymer like that. And if it breaks, and you've got two polymers like that, and they'll build more. If we seed it with that, it'll make a polymer like this. So we now actually have not only reproduction and variation, because there are two kinds of seed, two kinds of polymers, but we have heredity. Like begets like. However, we're not quite home yet, as far as the origin of life is concerned, or as far as living things are concerned. Because although we've got multiplication, variation, and heredity, we've got what I would like to call limited heredity. In this case, very limited heredity. There's only two kinds of things we can have. Although the two kinds of things breed true, that's no good for evolution. For evolution, we need an indefinitely large number of different kinds of things, each one of which will breed true. Until we have that, we really can't get the evolution of what is in this room, or evolution of the primroses, or of any interesting organism at all. So how are we to achieve not limited heredity with just a few different kinds of things, but unlimited heredity with an indefinite number of kinds of things? The answer to that question, in theory, I think, was first suggested by a physicist, a very distinguished physicist, Erwin Schrödinger, just more than 50 years ago, who suggested that you could achieve unlimited heredity if you had what he called an aperiodic crystal. Now, I'm not going to try to explain to you exactly what he meant by an aperiodic crystal, because I can, in a sense, do something easier than that by telling you that actually he was right, and that we now know what the aperiodic crystal is, upon which all life depends, and we know how it replicates. Now, in a sense, at this stage, I almost ought to apologize to you. I'm going to tell you how nucleic acids carry the heredity of life. I hope, in a sense, that most of you already know. My excuse for repeating it is that I do think it's certainly the most important discovery in biology of this century, probably the most important discovery in science in this century, and there is some excuse for repeating such discoveries, even if you already know them. The essence is shown in the next slide. I'm showing it for a molecule called RNA, not for DNA. For the purposes of this discourse this evening, the difference really doesn't matter. And that is my rather non-chemist's idea of an RNA molecule. It's a long string, and it's a string of 4 kinds of nucleotides labeled A, U, G, and C. And they have chemical formulae, and the fact that I don't remember their chemical formulae doesn't actually worry me all that much, because I was relieved to discover, reading many years ago, Jim Watson's discovery, I was going to say novel, it isn't actually a novel, his book, his account of the discovery of the structure of DNA. He admits that he and Francis didn't know the formula of adenine and cytosine and so on at the time, so I'm in good company. Anyway, these are 4 different molecules, and here are the isolated nucleotides, not all stuck together, which are supposed to be loose in solution, rather in the way that in that tube, before we put the crystal in, there were the units from which the crystal could be made. But here there are 4 different kinds of units, and the rule is a very simple one, that U pairs with A, that A pairs with U, A with U, U with A, but C pairs with G, and therefore C with G, G with C. And then all you had to do is to zip the thing together. It's a bit of a problem, actually, but that's all in principle you have to do. And then you've got not a copy of this, but a negative image of that, in the same way that a negative is the inverse of a photograph of the positive. But if we then proceed to copy this by the same mechanism, we'll get back to that. And that really is the essential process which underlies all of life. My friend Leslie Orgel, talking about this, once said that if you try to sort of trace back the history of life, back to its very origins, things sort of slough off one by one. You know, you lose legs and arms and leaves and things like that, and then you lose cells. Ultimately, you go back, and you're left with just the one simple principle of complementary base pairing, and this is complementary base pairing. And he said you get left with complementary base pairing like the smile on the face of the Cheshire cat. And I think that's right. It's really the root origin of the whole thing. Now, the thing I want to say about this is that one way of thinking about heredity, which wasn't obvious to us for a long time, but I think is obvious now, is to think that it's really just a matter of transmitting information. And as a result, I suppose, of the work of Wiener and Shannon at MIT during the war and others, we now have a kind of science, we have a mathematics of information, of how you measure the amount of information, how you transmit and encode information. The essence of that mathematics really rests in the notion that the amount of information in a message is simply to be measured by how many other different messages might have been transmitted and when. First sight, that sounds rather an odd idea, but you can illustrate it like this. You have your wife, your husband, your boyfriend, your girlfriend is in prison, and you receive a message, a letter from them, and the letter simply says, "I am well." How much information does that message give you? Well, it depends upon how many other messages he would have, or she would have been allowed to write. If the rules of the prison are that if you write a letter, it has to say, I am well, and that's the only letter you can send, then in fact the letter gives you what technically we would call one bit of information, and one bit only, that you're alive and not dead. If, on the other hand, it's a nice, modern, open prison in which you're allowed to do whatever you like from drugs downwards, then you could have written anything. Now the message, I am well, you could have written, I've got an attack of shingles, or, you know, I'm going to break out tomorrow, or all sorts of things. The message, I am well, gives you a lot of information. So we measure information in terms of the number of different messages we might have received. And when it comes to the message, the information in nucleic acid, the amount of information is simply 2 bits per nucleotide. It's simply the number of different nucleotide sequences of that length that might have existed other than the one you've actually got. Now, that's in a sense a digression, but I want to persuade you that for much of biology is today really permeated by the notion of information, and perhaps the simplest way is to remind you, or tell you, I'm not sure which it is, of some of the terms which are standardly used in molecular biology today. And I'll read them because I won't remember them otherwise. Translation, transcription, editing, code, message, reading frame, proofreading. All those are they're not loose analogies. Those are all technical terms. They have quite precise meanings. I was reading a book recently published by a friend of mine, I won't mention his name, it would be quite unfair, the other day, which contains the statement that bacteria don't have proofreading. Now, proofreading has such a precise meaning that I can assert that that statement is wrong. Bacteria do have proofreading. I say that not because I want to get at my friend, it's a very good book, but because I want to indicate this is a word which doesn't have a loose metaphorical meaning, it has a very precise meaning. You can say wrong. And not only does it have a very precise meaning, it means exactly almost what you would mean if you talked about proofreading. It means that when a cell, this is going on inside a cell now, replicates a nucleic acid molecule, having done all this pairing up, there's an enzyme that then goes along, group of enzymes goes along, looks at all these pairings, checks them, and if one of them is wrong, it takes it out and corrects it. That's exactly what you do when you do proofreading. And there's a special machinery for proofreading. So today, molecular biology is simply about information. Now, I want to be even more general than that and suggest to you that, in a sense, the last century, the 19th century, was a century in which science discovered how energy can be transformed from one form into another. From chemical energy into mechanical energy in a heat engine, in a steam engine, and from mechanical energy into electrical energy or vice versa, in a dynamo or an electric motor. This century, I believe, when we look back at it, will be seen as a century in which we discovered how to transform not one form of energy into another, but to translate information from one form to another. Though things never happen in neat centuries, of course, and the first machine which did this, well, I suppose strictly this is the 10th machine. Let me explain. This is Edison's phonograph. It's Edison's phonograph number 10. So I guess it's the 10th one he made. What it does is to convert the information contained in some grooves on this soft metal here into sound. It's not a terribly interesting sound, but it is a sound. Right? And, well, you can work out how it's working. Now, I guess that's not all that marketable. That's 1877. 11 years later, this was on the market. This, I'm not sure what this is actually called. Oh, it's on the thing. Here we are. The storage of information has moved from the metal to a wax cylinder. There's a groove on the wax cylinder. I don't think you can see it, but that's where it is. And on the label, it says, Edison's cold molded records echo all over the world. Yeah? And it does. Can you make it make a noise, Bryson? Can you hear the voice? There. That's the voice of Bill Coates, who was, I'm told, Bryson's predecessor in the guy who actually makes his lectures work, you understand. In fact, I met him. I gave one of these discourses 35 years ago, and he looked after me in the same way that Bryson is looking after me this evening. Now these are essentially the same machine, except that this one it's made of soft metal. Wax works a lot better. They have a property which is important to a biologist, and I'll come back to it in a moment, that they work both ways. We've shown it as taking the information out of the groove and turning it into sound. But if you shout like anything down there, you can cut the groove. I mean, that's how you get the information into the groove, right? So it works both ways. So does that one. You can shout down there and make a pattern on the wax. And that's how you make the pattern on the wax. Later, the thing became rather more refined, and we have here I won't say exactly a modern gramophone, but at least you don't have to wind it up. I mean, my first gramophone you wound up, but this one, yeah, let's have a listen. It's run by electricity, by something like that. Yeah. Okay, it's now saleable, you know, marketable, makes lots of money. But it's different from these other two in the following way. You can shout at that thing until you're blue at the face and you won't cut a record. Okay? This is a piece of information transducing machinery which transduces information in only one direction. Now, why does that matter? Well, I'll come to that in a moment. I just want you to remember those two bits of machinery. But to go back to biology, genetics is essentially the science about how information is stored and transmitted between generations. Developmental biology is the science of how that information is translated from one form to another during development, so that you finish up with fingers and ears and eyes and hair and stuff like that. And evolution, which is my own subject, is really the science of how the information gets into the animal in the first place. But they're all basically about information. Now, the first person to understand that, rather surprisingly, and I discovered that reading him quite recently, was writing, you know, like the machine, towards the end of the last century, not in this one, and it was the great German biologist, August Weismann. You'll know of Weismann, I'm sure, that he argued that acquired characters are not inherited. That is, that if you develop great big muscles, the blacksmith develops great big muscles, this doesn't mean that his children will have great big muscles unless they also exercise. And we all know that that's what he said. What is less well known, I think, is why he thought it. The essential reason why he believed it to be true is that he could not understand how it could work. He couldn't understand how the information, I've got big muscles, could be translated into some kind of chemical message in the sperm so that it would cause the child to have big muscles. Now, you may say, how do I know that that's what he was thinking? Well, essentially because he said so. In his big work he wrote towards the end of his life, The Theory of Evolution, he wrote it in German, but I don't read German, I can only give you the English translation. He wrote, if one came across a case of the inheritance of an acquired character, it would be as if a man was to send a telegram to China and it arrived translated into Chinese. I think it's one of the most astonishing sentences in the 19th century. He understands, by the telegram analogy, that he's talking about information, and he knows that the reason why acquired characters are not inherited is essentially the reason which was given a chemical meaning by Watson and Crick in the central dogma of molecular biology, that you can't, that the machinery for translating information works only one way. It resembles this gramophone, which is a machine for transducing information from a groove into a sound, but won't enable you to change a sound into a groove, and it doesn't resemble these two pieces of apparatus, which will translate information in either direction. So, in a sense, Weissman's statement about the non-inheritance of acquired characters is not a statement like saying you can't have a perpetual motion machine, or the speaker cannot now levitate. I mean, those are universal truths, even of this speaker. They're statements more of the kind that this particular piece of apparatus, as it exists in our bodies, doesn't work in both directions. Now, I said that everything depends ultimately upon the replication of nucleic acids. How far do we understand the origin of this process? Well, not by any means completely. Let me describe an experiment some years ago now by my friend Leslie Orgel, in which he started with a solution with the different nucleotides, the 4 nucleotides in solution, and he seeded it with a very boring RNA molecule consisting simply of a string of Cs. And he got out an RNA molecule in which, well, he got out the original RNA molecule and paired against the Cs G nucleotides. Except every now and then, about once in 10 or once in 20 times, the wrong one, a U, and then back to G again. We're getting complementary base pairing. Chemistry gives us that for free, in a sense. That is just chemistry. It doesn't explain the origin of the first replicating molecules, really for, I think, 3 reasons. First, it doesn't explain where the units came from, and these units are not among the easiest of molecules to imagine how they could be synthesized in the absence of living organisms in the primitive soup. Amino acids are fine. You know, you can make lots of amino acids without any organisms around to make them for you, just by shining sparks through the medium, but you don't get nucleotides all that easily. Secondly, it doesn't tell you how to zip the stuff together when you've made it, and that's presenting chemists with some difficulties, I think. The problem that worries me more than those two, because those are problems, they're real problems, but they're problems for chemists, certainly not problems for me. The real problem is the one of accuracy. You do make about 1 mistake in every 10 or 20 replications, whereas in your cells and mine, the rate of error is about 1 in 1,000 million, which is quite good, quite precise. How do you get from 1 in 10 or 20 to 1 in 1,000 million? Well, you get it by using enzymes. Among other things, you get it by proofreading, which depends upon enzymes and so on. What are enzymes? Enzymes are proteins. Enzymes are strings of amino acids. And this led to a very real problem. I mean, some of you may have been worried already, I hope you were worried by the fact that I talked loosely about RNA carrying a message, carrying information, and you may have said to yourself, well, it's all right saying that, but information means that it has a meaning. What is the meaning of this message? If it's just a message that replicates without having a meaning, it's not interesting. It isn't really information. Well, the meaning of an RNA molecule, the meaning is the protein that it codes for. The notion of a code, of course, also goes back to the early part of the last century, Here is a coding device invented, my wife tells me actually, in 1830 or thereabouts. The Morse code, and those of you who know Morse code will know that that is RI, or Royal Institution. So it works. You know, it'll code things. In just the same sense that is the code for Royal Institution, or RI, so particular sequences of nucleic acids are the code for particular kinds of proteins. And I want to spend the next quarter of an hour or so asking how that could possibly have come about. First of all, how's it done today in living cells? And it's a very complicated process. I can only describe it in a rather oversimplified form, and here it is. Along here, we have an RNA molecule with, I've shown them in triplets of bases. Those could be any one of the four, so you've got 4 of each, you've got 64 possible triplets along there. In some way or other, that is the code for a protein, which is up here. A protein is a string of amino acids, and you want to think of a protein as like a poppet necklace. You've got lots of little beads which fit together in what I learned as an engineer to call male and female fittings in order to produce a string. And you can fit them together in any order you like. Somehow or other, the sequence of these things along here, the bases along here, has got to determine the sequence in which the amino acids are formed along there. And it's done essentially, as I've shown it here, by a series of molecules called tRNA molecules, or transfer RNA molecules. They transfer the information from here to here, which have at one end of them exposed a little triplet. Our old friend complementary base pairing ensures that this pairs with that. This, needless to say, is called the anticodon, and that's called the codon. Attached at this end of each tRNA molecule is an amino acid. And depending upon what the triplet is down here, different amino acids are stuck on up there. And therefore, this tells which tRNAs to line up, and the tRNAs line up the amino acids. And then again, all you have to do is to zip together the poppets, and you've got a specific protein. And that's really all there is to molecular biology. You can well, very nearly all. But there is a problem here, which I once referred to as the catch-22 of molecular biology, or of the origin of life. The catch-22, you will remember, is that if, in Joseph Heller's novel, if you were in the American Air Force during the war, and you wanted to get out, as any sensible person would want to get out, because he was shot at by Germans. It was very dangerous and unpleasant. If you wanted to get out, the only grounds upon which you could get out of the Air Force was by proving that you were mad. But of course, if you wanted to get out, it proved that you weren't mad. So you couldn't get out. That's the catch-22. Well, here, the catch-22 of molecular biology is that you really can't have proteins unless you've got a lot of DNA to make them, to code for them. The messages down here, all the tRNA molecules and so on. But we've just seen from Leslie Orgel experiment that you can't have a lot of RNA accurately replicated unless you have some proteins to do it. So you're stuck. No proteins without RNA, no RNA without proteins. Well, we were stuck on this one for a surprisingly long time until it was discovered that, and I think it ought to have been discovered sooner, but it wasn't, that some RNA molecules can act as enzymes. Enzymes, catalysts, do not have to be proteins. And indeed, nowadays, you can hardly pick up a copy of Nature without finding an account of a new RNA molecule catalyzing a new chemical reaction of some kind or another. And this has led to the notion of an RNA-World in which, instead of what we have at the moment, a division of labor between one kind of molecule which does the information carrying, the nucleic acids, and another class of molecule, the proteins, which do all the work, all the catalyzing chemical reactions and so on, connected by the code, which is here, you only had the one class, RNA molecules, and that's fine. But nevertheless, we've still got to understand how the code originated. And before I take this slide off, I want you to notice what the essential thing is. And it's indicated by that red arrow there. Somehow or other, you've got to attach a specific amino acid to a specific tRNA molecule, and a different amino acid to this tRNA molecule, and a different one to this one, and so on. You've got to make specific linkages between particular tRNA molecules and particular amino acids, and that's the nature of the code. How did that originate? And what we need is the notion of the fitting of a particular amino acid to a particular tRNA. At this stage, I want to talk about the ideas of my young friend Szatmári, a Hungarian colleague, who has recently come up with an idea about the origin of the code, which seems to me, at least it's a jolly plausible guess as to what it might have happened, and it's also one which in the long run I think is testable, though at the moment I think it still has to be seen as speculation rather than as obvious truth. It's based on, the basic notion is that if you come across something very complicated like the code, and you can't see how it could possibly come into existence because it's so complicated, the chances are that in the early stages it was doing something quite different. Perhaps a classic example, if you look at the primary feather of a bird's wing, perfectly shaped and formed for flight. But, you know, poor old bird, if it wasn't perfect, you know, it would... So how are we to explain it? Well, there's no question the first feathers in evolution were not to help birds fly. They were to keep them warm. They were down feathers. And anything that sort of splays out of scale a bit is going to help to keep you warm. And in fact, one can think of a series of intermediates between no feather and a perfect primary feather. But the important thing is that the first function of feathers was different from their final one. And if my friend Ersch is right, the first function of this linkage, that linkage there, in evolution, was nothing to do with the coding for proteins. His basic idea is the following. Here is a ribozyme, which is simply sort of gobbledygook speak for an enzyme which is made of RNA instead of a protein. It's an enzyme which catalyzes something. Now, many proteins have what are called cofactors. They have attached to their surface, at their active site, some compound which is not an amino acid, which helps them to carry out some particular chemical reaction. Perhaps the most familiar example is the way that the hemoglobin in our blood, hemoglobin being a protein, can combine reversibly with oxygen, and that's what its function is, to take up oxygen and release it again, and it does it with the help of an iron atom combined inside the heme element, which is a cofactor working with the hemoglobin. Ersch's idea is that ribozymes would have benefited very greatly if they could use amino acids as cofactors. Because amino acids are chemically more reactive on the whole than the nucleotides. What he imagines is that particular triplets of nucleotides, and there are reasons why it would have been a triplet, were attached to a particular amino acid. Different triplets being attached to different amino acids. And then, if this ribozyme wants that amino acid as a cofactor, it has to have a tag at the end here of 3 nucleotides with which that one can base pair, and then the cofactor will attach. It's just simply to improve enzyme catalysis by ribozymes. Nothing to do with coding for proteins at all. But if it's a good idea to have one amino acid, maybe it might be even better to have 2 of them. And if 2, why not 3? And you're beginning to see how this thing might evolve into a message, into the modern method of synthesizing protein. In a bit more detail, his notion is shown here. Here is a cofactor an amino acid being attached to a trinucleotide by an enzyme. I'm not going to say synthetase, well I just did, but the reason I didn't want to is that I lisp, and if you lisp, synthetase is the sort of word that you try to avoid using. Anyway, there is a cofactor being made. It then binds to the ribosome, which is simply replicated in the way that RNA is replicated. And he imagines this evolving into a situation where this ribosome has become a message with a whole series of amino acids lined up against it. This thing here is another ribosome. It's the analog of what today is the ribosome, namely the body where proteins are synthesized, and in particular where they're stuck together end to end. And it's interesting that the enzyme within the ribosome, which today forms the peptide bond, sticks the poppets together, is, as a matter of fact, an RNA enzyme and not a protein. It's a kind of fossil relic left in existing living organisms to give us a clue to how it all might have happened. Now, I don't know whether that's right or wrong. It's, to me, the most persuasive suggestion as to how the code might have evolved that has yet been made. It has the enormous virtue that you can get from not having a code to having a code in small steps. Even being able to attach one amino acid out of the many different possible kinds to one nucleotide and using that as a cofactor for one of your ribosomes would be an advantage. And then if one, why not two? If two, why not three kinds, and so on. You can imagine a whole series of intermediate steps between not having a code and having one. But it may not be right. Now, I've talked in some detail about one particular change in the way in which information is encoded and transmitted. But I want to spend the rest of my talk suggesting to you that, in fact, one can see evolution, the whole of evolution, as punctuated by a small number, perhaps half a dozen, major transitions, major changes in the way in which information is encoded, stored, and transmitted. And my friend Sasmari and I have recently written a big fat book about this, but it really is, I'm afraid, a book which is written for people with a fair bit of biochemistry. Ursch always insisted in putting in more chemistry than I wanted him to, but since the ideas were often his, I couldn't argue. And I want just to sort of summarize what this notion may be. The first major event, of course, was the origin of replicating molecules themselves. A problem we have not yet solved, but which seems to me to be very close to a solution. I don't think we're all that far from having a full chemical account of the origin of the first replicating molecules. The next major change in the genetic system would have been the origin of chromosomes. A chromosome is simply a string of these molecules end to end, which has the property that when you replicate one of them, you have to replicate them all. No one of them can get copied without the others. The reason why that matters is the following, that if you have lots of different genes in the cell, let's say form a cell and you have lots of different genes, but each gene is separate from each other one. They're all competing for the same resources, but maybe some of them will replicate faster than others. And you'll finish up with just one of them that replicates faster than the others, and you've lost most of the information. They will compete with one another for the resources and nucleotides out of which they're made. But if you string them together in a chromosome, they have to cooperate. No one of them can get copied without all the others. It is, to use an analogy of Richard Dawkins, it's like 8 men in a boat. You can only get somewhere if you row with the other guys. I mean, it's no good trying to row in a different direction. So chromosomes, in a sense, forced cooperation on genes which otherwise would have quarreled with one another. The next major transition that we see having happened is the origin of the code, the origin of the situation in which information resident in nucleic acids could be translated into the sequence of amino acids in a protein, which is the basis of all development today. But it wasn't by any means the last major transition that one can see. Perhaps, I'm not going to give a full account, obviously, in 10 minutes I can't. But the next major event, I think, was the origin of sex. You perhaps don't think of sex as a matter of the transformation of information, but you should, among other things anyway. What happens in the sexual process is that information from two different parents is combined in a single descendant. And this enormously increases the amount of different messages, different patterns of information upon which natural selection can act, and therefore enormously accelerates the rate at which evolutionary progress can be made. So one's got the replication of molecules, the origin of chromosomes tying them together, the origin of the code translating information from one form into another. Then you have the origin of sex, enabling different messages from different parents to be combined. There was then a fairly long wait of a few thousand million years, maybe, before the next major invention, which was the origin of multicellular organisms. Organisms like ourselves, or like my primroses, which have not one cell with one copy of the genetic message, but many hundreds of millions of cells, each with a copy of the genetic message. Now, in a sense, you may say, well, that's not really an increase in information because they've all got the same information. And that's true, but they don't all use all of it. Some of your cells use some of the information. Bone cells use the information they need for making bone. Muscle cells use the information they need for being muscle cells, and so on. And a whole new signaling system, a whole new information transmission system had to evolve in multicellular organisms in order to ensure that different cells read different bits of their message. And we're at last, after a long delay, I think, at last beginning to understand how that happened. And it perhaps is the most exciting thing, actually, that is today happening in biology. The next major event in the complexification of life, making life more and more complicated, was a situation in which the unit was not a single individual or a pair of individuals and their offspring, but a society of individuals. This, of course, has happened in the social insects, in both the ants, the bees, the wasps, the termites, producing immensely complex societies, but it has also, and most notoriously, happened in ourselves. And to a very large extent, the fact that we have largely transformed the planet upon which we live depends upon that social relationships between us. But it's not by any means obvious what it was, first sight, that made it possible for us to form these very complex societies. But clearly something happened in our ancestors some 50,000 years ago, which changed them from a rather rare food gathering, possibly carrion eating, occasionally hunting animal, not an animal upon which one would have regarded, as wildly successful, into an animal which is enormously abundant and successful. It caused our ancestors to spread all over the world, to develop a complex material and social culture. It's hard to know for sure what that was, but it seems to me that there's only one thing really it could have been, and that was the origin of yet another information transducing machine, a new way of transmitting information between generations, namely human language. We transmit information from one generation to another, not just by our genes, though we still have that mechanism for transmitting information when needed, but we also transmit information from one generation to another, and from one person to another, of course, by the use of language. And there are, I think, good reasons to think, both on the one hand, that it was that invention of language which was the crucial event that took place in our history, which made us the dominant animal that we are, but also that event depended upon genetic changes in our own nature. In other words, that in order to learn to speak, we had to change genetically. But I want first to draw a rather tight analogy, if I may, between language and the code, the genetic code that I was talking of before, and the replication of nucleic acids. The point that I was trying to make in the first part of this lecture was to persuade you that there really is that the essential feature of the genetic system is that it can transmit not just a few messages, but an indefinitely large number of messages. There is another coding system which will do that, and that is human language, but peculiarly human language. Animals do have languages. They have words. A vervet monkey has a word for a leopard. It has a different word for an eagle. It has yet a third word for a snake. It has yet another word for another troop of monkeys. And when it uses those words, other monkeys understand what is being said. So it's not that they have no language, but they have a very limited number of things that they can say. Very limited indeed. We can say an indefinitely large number of different things. And the thing that differentiates us from the vervet monkey is that we have grammar and they do not. You see, we do not have 4 different words to describe a sleeping lion, a running lion, a sleeping dog, and a running dog. We have a word for lion and a word for dog. We have a word for running. We have a word for sleeping. And by combining them together, we can increase enormously the number of possible things that we can say. If we have 1,000 nouns and 1,000 adjectives, then we can say 1,000,000 things. Not just 2,000 things, which would be all we could say if we didn't have the grammatical notion of predication. But of course we have a lot more grammar than just the notion of predication, that's to say, of ascribing properties to objects. We have highly complex grammatical skills, universal skills, all human beings have them, and the dramatic thing, to me at least, about grammatical skills, and the thing that makes me believe that they are in some sense genetically programmed skills, is that in speaking we make use of rules of which we are unconscious and which we couldn't describe if we were asked. And let me, to persuade you, let me say 2 sentences to you. How do you know what he had for dinner? What do you know how he had for dinner? The first sentence is, I hope you'll agree, grammatical. The second sentence, again I hope you'll agree, is not. I suspect that unless you happen to be a trained linguist, and maybe not even then, you have no idea whatever what the rule is that tells you the first of those 2 sentences is grammatical, and the second one isn't. You have no more idea how you talk grammatically than you do how you digested your dinner. You really don't. And that makes me feel, and there are other evidences as well, of course, that this linguistic skill is something which required genetic evolution specific to humans, not to cause us to speak English rather than French, but to to enable us to learn the grammar of the language with which we were brought up. I almost said that this was the last of the major transitions that have happened in our evolution, but of course it isn't. There have been 2 further major transitions in the way in which we transmit information between generations. Some 5,000 years ago, we invented writing. And having invented writing, we could construct and live in immense complex societies with big cities and taxation systems and that kind of thing. I mean, I suppose really taxation is the most important consequence of writing. But even that is not quite the last revolution. The last revolution, I suppose, is this. This is a CD. Nowadays, if you want to make horrible noise, you don't buy one of these things, good old-fashioned 78, you buy one of these. This thing, although much smaller, contains as much information as about 100 of those, roughly. In other words, we are now storing information electronically, not in brews on wax. We are transmitting it electronically. And maybe the future lies with entities which transmit information electronically rather than otherwise. When I look at my two grandsons playing Sonic Hedgehog on a computer, I wonder who is controlling whom. You know, at first sight you might say the child is controlling the computer, it's pressing the buttons. And so indeed it is. But I think if a Martian were to look in and look at what was happening, he'd say that computer is controlling that child. But it doesn't seem to me to be wholly impossible that the future lies with reproducing, self-replicating entities which are transmitting information between generations, not by DNA, not by, as I'm doing it now, by human language, but by electronic means, and that we shall either be their servants or be totally expunged as no longer strictly necessary. This is a bit alarmist, perhaps, but at the moment we are, in a sense, saved by the fact that this machinery, although it does carry information, although it transmits it, these machines are not self-replicating. They still do require us to build factories in which they're made. They don't build their own factories. They're not completely taking over. Maybe this is silly. It may well be. I don't have any doubt in my mind, however, that the impact of electronic storage of information, electronic transmission of information, is going to be as profound as the other earlier transitions that I've talked about, the origin of replication itself, of molecular replication, the origin of the genetic code, the origin of sex, and so on. We are, I think privileged is perhaps not quite the word, we are in fact living through a final revolution in the way that information is transmitted, stored, and encoded. And fortunately, I don't have to tell you the results because I've been saved by the bell.